Good morning, everybody. I will give two lectures today, and we kind of decided to connect them also to each other because they are well, from the biological topics they are different, but from the data analysis they are kind of similar. And so the first one is work from Rong Xu, and we did a lot of nanopharmacological force sensing, and for this it really requires to have a very distinct and detailed data analysis, which I will also go into. We are talking about monoamine transporters, and they belong to the amine carriers. And I will talk first about the serotonin transporter and then the dopamine transporter. And they are medically highly important because they are connected to a lot of diseases actually, like for instance Parkinson, ADHD, depression, autism, and so on. So they are a highly medically important target. And this is what they are doing. So it's actually pretty much the same with serotonin or dopamine transporter. So they are sitting in the neurons at the end of in the presynaptic neuron. And what they are doing, they are responsible for the reuptake of monoamines like serotonin or dopamine from the cleft here, from the synaptic cleft. And they are responsible because monoamines are secreted from the postsynaptic neuron, and the monoamine transporter like serotonin and dopamine transporter are responsible for the reuptake. And what happens if they are blocked, and they can be blocked by drugs, then the concentration of the monoamines in the synaptic cleft is high, and this induces a feeling of happiness. So it's in one case it's important for depression, for instance. You want to keep happiness up. On the other hand, it's also how cocaine acts. So cocaine blocks the monoamine transporter, and this induces this feeling of happiness. So you can use them, and you can also misuse it, depending on if it's medical. But then, of course, the development of the medicines is also highly important because it can easily lead to addiction, and that is the key for developing the medicals. And this is a transport cycle, so it's a secondary active transport cyclist. ATP is not involved in that, that's why it's called secondary active. It's basically regulated by a sodium gradient. And the transport cycles can be depicted in 4 steps. So in the first step, the transport is in the outward facing position, so all the transport is accompanied by heavy conformational changes. So it differs to ion channels where there's just minor conformational changes. The transporters are exposed to severe transformational changes during the transport cycle. So in the first step, the substrate and so like monoamines and sodium are bound in the outward facing state. Then it goes into the outward facing occluded state, so it kind of closes up. Then it goes into the inward facing open state, and then the substrate and sodium are released. And then the next step is the inward facing occluded state before it goes back there. Okay? So this is how you can imagine the transport cycle. And there has actually been heavy discussions about the binding sites of the transporters. So it is most X-ray and cryo-EM studies of the monoamine transporters show only one binding site of the substrate, of whatever substrate. And there is example bacterial LOT, so these are kind of a little bit analogs, but this is also from the human serotonin transporter. So they show only one binding site. This is a kind of central binding site, and with the high resolution, you know which amino acids are involved and so on. But then, on the other hand, some computational studies predict the second binding site, which is which is a vestibular binding site, a little bit outside. So we are talking about central binding site, which is the S1 site, and vestibular binding site, which is the S2 site. So please keep this nomenclature in mind. So, and this is of course a discrepancy which we aim to resolve with single molecule force spectroscopy. So what we did is we bound the drugs to the AFM tip, and then we performed force distance cycles, you know this movie already, on living CHO cells in which the monoamine transporters were expressed, were highly expressed. And you already know that, so if this is a trace, this is a retrace. Here you see a little bit of shift in the trace and retrace, which is characteristic if you have a soft surface and if you do it a little bit faster. But still, of course, we can depict the unbinding forces from the retraces, which goes like this. And what you actually also see is that the length, unbinding length, is rather high now. It's in the range of 300 nanometers. Yeah? Before that, we had something like 20 or 30 nanometer because of the PEG spacer. And the reason for that is that the cell is very soft. Yeah? So what we pull up the cells by 200 nanometers because it's so soft. Okay. And also the tip chemistry is quite tedious with this because they are small substrates. Yeah? And so you can see that here the first example is on S-citalopram, and we are just also a little bit lucky because we cannot. You have to kind of newly synthesize these drugs with a tag on it. Yeah? So they have to be biochemically active, and we are physicists. We couldn't do that. Yeah? But this is a great collaboration with Amy Newman from NIH, and she's doing fluorescence experiments with those drugs. And of course, you also have to put on a fluorescence label for that. Yeah? So she sends us the drugs, she binds the fluorophores onto it, and we are binding the drugs to the AFM tip. And this, so she synthesized it with an NH2 group, but then you can bind with a pentyne NHS ester, and this makes it biochemically active with click chemistry, which is a very strong chemistry. Yeah? And of course, also this functional group should be opposite to the binding epitope to the transporter. Yeah? Otherwise, it would interfere a lot with the binding. Yeah? So that is really kind of arts of medicinal chemistry, which we cannot do, but which we do in collaboration. And then we end up getting a tip that is functionalized with the drug. In this case, S-citalopram, and S-citalopram is something like a cocaine analog. Okay? And then what we actually found, if we do our force spectroscopy experiments many times, we found two classes of dissociation forces: weaker forces and stronger forces. Yeah? And our anticipation was that the weaker forces come from the binding to the S2 site, to the vestibular binding site, and the stronger forces come from binding to the central binding site. Okay? But the question is, okay, how to prove that? Yeah? And this is how we show. So, as you know, we have to do these experiments many times, so like 1,000 experiments, and then we construct the experimental probability density functions. Yeah? You remember we just add up Gaussians with unitary area, and the width of the Gaussian corresponds to the thermal vibration of the cantilever. And if we add up all these Gaussians, we get a histogram, a purely experimental curve again, with two maxima, yeah? Not with one, with two. Yeah? And this maximum belongs to the weaker forces, and this one to the stronger forces. Okay? So this is what we get. And you can also see now here again the importance of these experimental probability density functions, because you really need now a high resolution to distinguish between the forces. Okay. And then we did a mutation in the vestibular binding site, a point mutations, and you can see that we lose the maximum of the low forces. Yeah, and so with this, and since this, it's not. I mean, the structure is known, of course, and since this mutation has been done in the vestibular S2 binding site, we know that the weaker forces come from the vestibular binding site. Okay. Of course, we also did Bell-Evans model. You just see the fits here because if I show all data, it's too it's too complicated. But yeah.
Reverse also is possible to block S1 and just keep the S2, which is lower.
I will come to that. That's a very interesting point. Yeah.
S2 is decreasing the attractive forces between the.
Yeah. Well, if we produce a point mutation in the vestibular binding site, we lose the weak forces. Yeah. You can see that here, and this means that the weak forces come from the vestibular binding site. Okay. Yeah. You don't agree? Okay. Okay. And you see that, and of course, what we get now in the Bell-Evans distribution is two curves. Yeah, one for the weaker forces, one for the stronger forces. And this is, and if we do the mutation, we only get the stronger forces. Okay. So then we exchanged sodium to lithium, and what we see now, if we do that we now use the maximum of the stronger forces. Yeah, which means that the S1 site is sodium dependent, but S2 apparently not, because we still have the maximum of the weaker forces. Okay, so only the S1 binding site is sodium dependent, but not the S2 binding site. And it is also interesting to note here that lithium is actually also used as a medical, yeah, against depression, for instance, because it destroys, it blocks the binding. Yeah. Okay. Good. So there are actually two enantiomers of citalopram, the S-citalopram and the R-citalopram. And they are known to induce, in therapies and more in vivo experiments, to be different in the effect. So I will show you that the two binding sites are allosterically coupled to each other. Yeah? So let's first talk about allostery. You have a positive allosteric effect if the so first of all, you need two binding sites, yeah, for allostery. And the positive allosteric effect if binding of a ligand to site 1 enhances the binding activity to site 2. Yeah? This is a positive allosteric effect. And the negative allosteric effect is if the binding of ligand at site 1 reduces the activity of binding at site 2. And now we come to the experiment that Shiva mentioned, which shows a very interesting effect. Because, so we put now 30 micrometer of S-citalopram into solution. Yeah? This is, this is a high concentration. So what we would expect, and we would have expected, is that we block the binding. Yeah, it's as a usual blocking experiment, what I also showed before. Yeah? If you block the binding site with the drug, then the drug on the tip cannot bind to the transporter anymore. Yeah, but no, it was different. You can see. So this is the binding curve without citalopram in solution. It looks like this. And now, when we add citalopram in solution, we lose binding to the S1 site, to the stronger binding site. This is what we block. But we retain the binding to the weak binding site, but the forces are shifted to higher forces. Yeah? Okay? So we are not able to completely block both binding sites. So this is with the S citalopram on the tip and the R citalopram on the tip, with both of them. Okay. So apparently we block the S1 side, but then the binding forces increase to the S2 side increase. This means we have a positive allosteric effect here. Okay. That is with the S citalopram in solution. And then with the R citalopram in solution, we see for R citalopram on the tip that actually also the forces at the vestibular binding site are shifted to higher forces. So this is again a positive allosteric effect, but with the S citalopram on the tip, the forces are slightly shifted to lower forces, which means this is a negative allosteric effect. Yeah? So we have a coupling of this both binding sites. And it's now also the question, how are they coupled just in solution? And one hypothesis is that binding, because apparently they cannot bind at the same time to both binding sites, so that maybe the binding to the central binding site occurs through the vestibular binding site. Okay, so that there's a transfer. Okay. Yeah, that's a good question, but the affinity to the central binding site is much higher. Yeah, so it would first bind. So what we block must be the central binding site, not the vestibular binding site, because the affinity is 100 times higher to the central binding site than to the vestibular binding site. Now, what we can really assume that if we add the citalopram into solution, that it would first bind to the central binding site, because the affinity to the central binding site is 100 times higher than to the vestibular binding site.
So with the wait time, like with the contact time, can you change the probability like PDF?
What changes is so you can deduce the kinetic on rate, the probability of binding, but it doesn't change the forces.
Right, but the probability of binding to the S1 would increase.
To the S2 would increase, yeah.
The S2.
Yeah, because the S1 is blocked in this case.
Not in this
Not in the other case.
Why is that?
This is I mean, if you look at the probabilities, if you look at the PDFs, you see that the probability to the S2 binding site is here a little bit higher. But it's a little bit random whether it binds to the S1 or to the S2. Yeah. So from the kinetic on rate, there doesn't seem to be a lot of difference between from S1 and S2.
It's also similar RBDs from the corona thing. The on rates didn't change between binding to one or two. Like in case of corona.
No, I think that's now really completely different story, because here you have two different binding sites. With the RBDs, they all had the same binding site, because for all of them was to ACE2. So this is why it would also be wrong to fit the Williams model here, yeah, because it's not a double binding. It's binding to either this or other that, and the two are different.
So if we use lithium ion solution, we won't get that peak, single peak.
Well, with lithium, you can see that here.
Because for lithium, S2 is involved, right?
Right. So
S2 is blocked.
it is more actually dependent on sodium, yeah, because you could also exchange sodium with something else, and you still would lose. So if you don't have sodium in solution, you don't measure strong forces. Yeah. So the central binding sites, binding to the central binding site, needs sodium. This is more the message. And one possibility is lithium, but if you substitute it with another salt than lithium, you still would lose the strong forces. Yeah, and we actually also compared. So first of all, this single molecule force spectroscopy is really then the only method which can identify two binding sites. Yeah, you don't get it with BioCore. Yeah, but we also compared it with a different method, with a electrophysiological method where you can, I don't want to go into detail, but you measure the capacitance somehow, and then you can block the current by adding a citalopram. And the kinetic off rates were very similar. And first of all, you can also, just as a proof that we are measuring the right thing, but you can see now also the difference for the kinetic off rate of the S1 and S2 side. And you can see that it's kind of a factor of 50 that the S1 side has a higher, a stronger binding. That is a factor of 50. And you can also see that there's a difference between the R-citalopram and the S-citalopram. So the S-citalopram has a much higher, well, in this case, kinetic off rate, but we can also talk in terms of affinity. Okay. So we can really get here now from the interaction energy landscape, we can identify a kind of transient binding sites because of the sensitivity of the method. And we have two binding sites that are allosterically linked. Substitution of sodium with lithium disrupts the S1 binding site, and if we mutate a single point mutation, disrupts the S2 site. And 3 months after we had identified two binding sites on serotonin transporter, a Nature paper came out which also revealed the two binding sites in the crystal structure. Okay? Without citing us, of course. Because what is force spectroscopy? Okay. But not only one can reach the, on the cantilever there's more, yeah, but this is but only one can reach it. So we don't see double binding, because if it would be two, yeah, the concentration of the serotonin transporters is high, then we should also see sometimes double binding, in particular on cells, sequential disruption of two.
Then it's self-evident that you see those with only one binding.
Yeah, exactly. Yeah, right. Yeah, exactly. Yeah. So we only see one, but either high force or low force. Okay. And well, about the same question as for the serotonin transporter, came up with the dopamine transporter, which in X-ray also shows just one binding site, but there's some experimental evidence like linking two dopamines with a linker and reaching higher affinities, that there could also be two binding sites. And dopamine and serotonin transporter are relatively similar with the structure, just with the difference that SERT transports serotonin, DAT transports dopamine. So we were asking the same question, and we also bound MFC12 to a tip. This is also a cocaine analog. And first of all, what we saw is that zinc, for the dopamine transporter, zinc is important for its function. So the binding activity is zinc dependent. And then we also bound dopamine to the AFM tip. And we also found that so we can also see that we have here a sodium-dependent central binding site and also a vestibular binding site with dopamine on the dopamine transporter. So about the same, also a central binding site and the vestibular binding site. And it is also sodium-dependent. You can see if we substitute sodium by NMDG, in this case, we lose the strong interaction forces but retain the weak interaction forces. Yeah? So also for the dopamine transporter, the central binding site is sodium dependent. And also that the stronger binding originates from S1. But then, of course, we wanted to again localize the binding sites, so we did several mutations. And this is a mutation here, a single point mutation, and this point mutation now destroys the central binding site. Yeah, so we know this is a mutation in the central binding sites, and we confirm that these mutations, because we are losing the strong forces, is now that the strong forces belong to the central binding sites. I mean, you can see there's still a little bit here, yeah, but this is biology. Biology is not digital, so this means it just decreases the probability for binding strongly. And then other mutations, so with the lysines, two lysines mutations, you can see that we are losing the binding to the S2 site, and this means also that's the proof that the weak forces come from binding to the S2 site. Okay. But what we don't have with the dopamine transporter is an allosteric effect. Yeah, I mean, it's again similar if we inject so either MFC12 to 12, which is a cocaine analog, or dopamine in solution. We block the S1 site, yeah, but not the S2 site. But the forces of the S2 site do not change; they remain the same. Yeah? This means that it can still only bind to one side, but for the dopamine transporter, it seems that S1 and S2 are not allosterically coupled. Yeah, so basically, if we block the S1 side, the S2 side doesn't care. Yeah? It's not influenced by the binding to the S1 side. Yeah? Okay, but two binding sites are still not discovered for the dopamine transporter in X-ray crystallography. Yeah? And the question is why? And this is now a little bit of speculation, but it's also kind of nice to speculate a little bit in science. If we look at the forces now, for serotonin, so here, yeah, if we look at the different forces at a certain loading rate, at 50 piconewton, so the S2 site of that has a relatively weak binding, which is below 10 piconewton or slightly below for dopamine and for the MFC12. Yeah. Then this is the binding to the S2 site. Binding to the S1 site is stronger. You can see that for dopamine here and here. But then the binding to SERT of escitalopram is in the range of 13 piconewton. So that is higher because of this positive allosteric effect. Yeah? And this stronger binding may be right or not, but there's a correlation. Yeah? May be responsible that the S2 binding site has been discovered in SERT, but not in that, because the binding forces, the interaction forces are stronger. Yeah? And it seems to be also very difficult to bind it into solution, because we also don't block it. Because it's really high concentration of the drugs. But we only block the S1 side and not the S2 side. So it seems to be quite. But it might be transiently, as I said. There's no proof, but it might transiently go from S2 to S1. Yeah. Yeah.
So
Yeah. There is. Yeah, but they show only one binding site. Yeah, I mean for SERT now they also show two binding sites after some effort. Yeah, and after we published it, it also shows two, and there is a clear idea that also from other data, not only from force spectroscopy, but they are kind of indirect, yeah, and from modeling and whatever. And there's clearly the suspicion that there's also two binding sites. It seems to be clear, but so far they couldn't prove it with X-ray crystallography or electron microscopy. So far there's no proof.
binding sites which are not well exposed. So that could be a challenge for AFM to find out these binding AFM molecules.
Yeah, but it works. You measure binding to it.
What if there are structures whose binding sites are not exposed?
But if a binding site is not exposed, then it's not a binding site, because then nothing can bind. Yeah, so it must be exposed, because otherwise, if it's something inside which has in principle an affinity but is not accessible, then it cannot act as a binding site. So this is, I would say, because. Okay. Yeah, so there is also now we showed that there's 2 binding sites in DAT for both MFC212 and also dopamine. And we could also localize them by many mutations. So mutations in the center of the transporter disrupt this one binding site, and then also it requires also the S1 binding site of the dopamine transporter requires sodium, and we can also disrupt the S2 binding site with certain mutations. But there's no positive allosteric effect to S2, and this might also be the reason why it has so far not been proven with crystallography, because it might be that the the binding to the S2 site is so weak that during the whatever crystallization process you lose it again if you have it. Yeah, and the paper has been accepted now a little bit more than a month ago. And it was a collaboration with many people. So also other institutions, physiology and pharmacology, medical university, and as I remember, very important, the collaboration with Amy, who is synthesizing all the drugs for us. Okay. All right. Okay, thank you very much.